So good afternoon, everybody, and welcome to this ELIXIR webinar. So today I'd like to share with you our work and that of our partners on this particular ELIXIR implementation study that looks at developing a microbial metabolism resource for systems biology in Europe. So the work I'm going to talk about today was performed in collaboration with the group of Claudine Medigi at GeneScope and the French Institute of Bioinformatics, as well as the Ensembl Genomes group at Ensembl EBI, led by Paul Kersey. So this webinar is divided into three main parts. So first we'll look briefly at why microbial metabolism is so important for human health and economic well-being. We'll also look briefly at one of the main tools for the study of microbial metabolism, which is the genome-scale metabolic network model. Then in the main part of the webinar, we'll look in more details at the objectives we set ourselves in this implementation study. And broadly speaking, this involves looking at ways to better support the study of microbial metabolic systems using computational methods. And we'll look at some of the results we obtained. Finally, we'll round off the talk by mentioning some of our future plans, not only of the people involved in this implementation study, but also the wider community, which includes actually developing an ELIXIR community around this subject, the subject of microbial metabolism and biotechnology. As Promisil said, this webinar should last around 30 to 35 minutes, and we'll take questions at the end. So, there are many motives for studying the metabolic processes of microbes, as these can impact human health and well-being in a wide variety of ways. So microbes are common pathogens. We've all suffered from microbial infections at one time or another, and drugs that target microbial metabolism are among the most commonly prescribed drugs of all, and many antibiotics actually target specific metabolic pathways in microbes. One of the consequences of this is that the development of resistance. So identifying the differences between human and pathogenic metabolic systems, and identifying targets which could inhibit microbial growth, either alone or in combination, could really be key to developing new drugs, especially for strains that resist traditional antibiotics. Another area of great interest is understanding the impact of commensal microbes on their host. So the human microbiome. So each human being contains several kilos of microbes, and these microbes and the metabolites they produce can directly affect human health, sometimes in surprising ways. So recent studies have tracked metabolites like short-chain fatty acids, which can have an impact on health and obesity and metabolic function from the mother's gut through her milk and even into the infant. So the microbial content of the mother and her microbiome can directly impact the health of the child. Another area of interest is the use of microbes as factories. So microbes have an enormous commercial potential, so they can be engineered to produce desirable chemicals like drugs and biofuels. Those are areas of particular interest, or also break down undesirable ones like pollutants such as plastic. And there's a lot of interest in developing bacteria that can attack these kind of micropollutants and microplastics. Finally, microbes are a great source of new tools for doing biology and chemistry. There are billions of species of microbes on planet Earth, and only a tiny fraction of these species have been characterized at any level. And this vast genetic diversity presents an enormous potential reservoir of, for instance, enzymatic activities which could be exploited commercially in industrial processes. So one well-known example is cellulase, which can be used to generate biofuels from plant materials. And estimates vary, but they're in the range of billions of dollars for the market potential in the US alone. Okay, so the study of microbial metabolism is of wide interest to many areas of human health and activity. Now, the tools that are used to study microbial metabolism are, like microbes themselves, evolving rapidly. We've seen a real explosion in recent years in the use of computational approaches, particularly computational modeling of metabolic systems to study them. So, of course, these kind of approaches are generally applicable to all types of biological system, from metagenomics communities all the way through to eukaryotes, and are not restricted to individual microbial systems. So it's these computational models which are really the focus of this implementation study. Okay. But before we get into the fine details of how these computational models are constructed and the work we did in this study, we'll just illustrate the main features of these models using a cartoon example. Okay, so this is our cartoon computational model of metabolism. It's very small and relatively simple compared to a real model, which would be expected to include thousands of individual reactions. Okay, so each of these blue arrows represents a metabolic reaction, which describes the interconversion of some substrates and products. So in addition to this metabolic network, we have a variety of other features in the model. We have a set of inputs which define the growth media. We have a biomass, or which is the output. This is kind of like an objective function. If we can produce biomass, then this is a viable model. The model can be compartmentalized to some degree. Here we have a single compartment represented by the cytoplasm, but we can also have models with multiple individual compartments, particularly for complex eukaryotes. So we can ask a variety of questions of these kind of models. We can calculate the fluxes through the individual reactions and study how the model behaves under a variety of conditions. Okay. So for instance, we can ask, can the model grow? If we calculate the fluxes through the reactions, do these fluxes support the generation of biomass on a particular medium? So we can also vary the media. We can add or remove elements and define the optimal or minimal media for the growth of particular microbes of interest. We can also engineer the system. We can graft on additional reactions or entire pathways which will allow us to produce compounds of interest, like, for instance, drugs and fuels. And we can also ask questions about which are the essential genes, either to produce biomass or these individual products. Okay, and this is obviously of interest in identifying drug targets. We can identify not only individual genes, but also combinations of genes which can effectively block growth. So these are some of the kinds of questions you can ask with these sorts of models. As I said, this is a cartoon computational model of metabolism. The real models contain thousands of enzymes, reactions, and metabolites, and they're derived from real genome annotations. Okay, so that's a cartoon example. So if we look at how real models are actually built, so we begin with an annotated genome, and from this we create a draft metabolic network. So the first step is to identify the genes in the genome. So we have a variety of genome annotation pipelines to achieve this. And once we have identified gene sequences, we can infer protein sequences, and then using normally homology-based methods, assign putative functions to those enzymatic activities. These enzymatic activities are then mapped to reactions, and the reactions specify how metabolites interconvert. And these, as I mentioned in the previous slide, can be compartmentalized. So the generation of a draft genome scale metabolic network is very much linked to genome annotation. Okay? Following this, there's usually one or more iterations of refinement of the draft model. This can involve manual curation or a variety of computational approaches. One of the common activities at this step is to add additional reactions which fill gaps in the draft metabolic network. So we might be missing genes in our genome annotation, or we might have functions which are performed by non-homologous enzymes, and these might be missed in the initial pipeline. So there's usually several iterations of refinement and curation to generate a useful network model. And then once we have a useful network model, we can begin simulations and comparing the results of those simulations to experimental data. Some of the questions, addressing some of the questions which we looked at on previous slides. Can the system grow? What is the effect of deleting particular genes? What are the media requirements to get optimal growth? And how can we graft on pathways to synthesize or degrade chemicals of interest? So this is a general schema for how computational models are built in schematic form. Okay. Now, the objectives of this particular implementation study were really to look at how our resources, the resources of the ELIXIR partners involved in this study, could be integrated to better support these kinds of activities. Okay. So This involved looking at issues of data interoperability between the systems. So we spent some time identifying common standards, which can be used to describe the biological entities in these models. That includes common ontologies and identifiers, all the way from the genes in the genome to individual metabolites. And we also looked at components that are missing and require development, like, for instance, a database of gene-protein reaction links, which are known to occur in models, but which are missing from knowledge bases. Okay, so knowledge bases deal with knowledge, and models often include inferences or hypotheses, which are not found in these individual knowledge bases. But yet they could still be useful for people building models. Also, there's a wide range of specialized tools which are useful for building and also reconciling different models. Now, the focus really here in this ELIXIR implementation study was looking at how our resources can be integrated to better support the generation of metabolic models. So I'll just tell you something about the resources involved. So the three partners, ELIXIR Switzerland, ELIXIR France, and Ensembl EBI, brought 5 individual resources to this implementation study. The first of these is Rhea. This is a curated knowledge base of biochemical reactions, and it's built on the ChEBI ontology. Okay, so that provides information about metabolites and about reactions. The second is HAMAP. This is a resource for the classification of protein sequences by homology and their functional annotation. Okay, so it's also integrated in other resources like Interpro, for instance. So HAMAP can provide enzymatic functions for protein sequences. MetaNetX is the third resource, and it differs from the others in that it's been specifically designed to support the generation and analysis of genome-scale metabolic networks and models. Okay, so it's not a knowledge base per se; it's more of a tool for the creation and manipulation of models. MicroScope is a platform for microbial genome annotation and curation, so that provides gene and genome information and functional annotations. And Ensembl Bacteria is also a genome annotation system or resource with over 40,000 bacterial genomes. So these are the resources which we included in our study of microbial metabolism. So these partner resources, they map pretty well to various parts of the generic modeling pipeline. So Rhea provides information on metabolites and reactions. HAMAP provides information on enzyme functions. And MicroScope and Ensembl Bacteria provide a range of information all the way from the genome sequence through the protein sequence to enzyme function. So we cover most of the requisite data types for building genome-scale metabolic networks. The aim is really to see how to integrate these resources better to more effectively produce them. As I mentioned, MetaNetX is designed specifically to support the generation of models. And one thing that this resource is very good at is providing annotation for the models. Okay, this is something that we studied quite in detail as part of this ELIXIR implementation study. One of the things which really complicates the use and reuse of models is the degree to which they're annotated. Okay, so unlike knowledge bases such as Rhea or UniProt or others, the process of model generation is something which is often carried out in research laboratories. Okay, so it's standardized to varying degrees. So to illustrate this, I'll just show you one of the main features of this MetaNetX resource. So underlying the MetaNetX platform is a common nomenclature for metabolites and reactions. Okay, so it's a common namespace called MNXREF. And this namespace is created by merging metabolite data from a large variety of resources, including ChEBI, which is included in this implementation study, but also many other resources worldwide, such as, for instance, Metapsych in the US, KEGG in Japan. And it includes not only knowledge bases, like those I just mentioned, but also modeling platforms, like, for instance, the BIG modeling platform, which is maintained in the US, and also SEED. Okay, So this common namespace is really built by reconciling metabolite data from all these different resources, and that's achieved using a variety of different kinds of evidence. So this colored figure, it actually represents a reconciliation pathway to generate a single metabolite identifier, which can be used to bridge all these various resources. So each of the metabolites is represented by its name and an identifier, or identifier in text, and each of the colored circles represents a reconciliation event. So if we look at the top right of this figure, we can see a ChEBI metabolite is being reconciled or merged with a metabolite from Metapsych. Okay, that's the pink circle, and these are merged because the source databases provide exactly the same structure for these two chemicals. Okay, and structural reconciliation is achieved using INCHI keys. Following this, these two metabolites are then merged with a compound from lipid maps, which has again the same structure, but it differs in the charge state. Okay, so if you look at the color key and the reconciliation pathway, you can see the stepwise reconciliation of our metabolites, how it proceeds at gradually lowering levels of specificity. Okay, so because this reconciliation process includes resources, ontologies like ChEBI, and also the namespaces used in modeling platforms, it provides a way to bridge metabolic models and commonly used ontologies and knowledge bases. Okay, so to illustrate this, I'll just show you how a model is annotated in the MetaNetX pipeline. So we'll illustrate this with a yeast metabolic model, which contains around 1,500 reactions and 900 genes. Okay. So if we look at the underlying data present in this model, this is a single reaction from the model in SBML, which is Systems Biology Markup Language. So we see a list of reactants and a list of products, and these are represented using text labels. So the M at the beginning of the label specifies that this is a metabolite. The C at the end says this metabolite is in the cytoplasm, and the intervening abbreviation gives the user the nature of the metabolite. Okay. So the annotation of metabolites and reactions in SBML from different providers varies. So different providers might use different labels for the same metabolite, or they might or might not provide links to databases. So what happens when we pass this SBML through the MetaNetX pipeline? We annotate these metabolites with standard identifiers for these entities and their compartments. So we get ChEBI for metabolites, and we get gene ontology terms for the compartments. Okay. Once we have annotation of metabolites, this metabolite annotation is then used to infer links to reaction databases such as Rhea and the enzyme classification. So MetaNetX is providing essentially post hoc annotation of metabolic networks and models. What we'd really like to do in the future is to support the generation of better annotated models at source, help people to provide annotations in the models which facilitate their reuse by others. Okay. So we're really focusing on the generation of a draft genome scale metabolic network. How can we provide high quality functional annotations for these networks using the resources that we have at hand? And there's two aspects really to this. The first is standardizing the semantics amongst these various resources. Are we using the same terminologies to describe the various biological entities? And the second aspect is the data models. So are we using data models that are interoperable amongst these various resources? Okay. So to achieve these goals, we used RDF and related technologies, and I'll show you that in more detail. Okay. So for those of you who are not familiar with RDF, it's one of the W3C semantic web standards. The semantic web, as probably most of you know, is a web of data that's designed to be processed with minimal intervention, really by machines. So you could say that a lot of the work here is about making our data resources more fair in terms of metabolic network construction. So RDF is a data model, and I'll also talk about SPARQL, which is a query language to ask questions of that data model. Okay. Now, within RDF, a resource is anything that can be identified by a URI, and there's an example URI on this slide for a protein from UniProt. Okay, and RDF is composed of triples. These are a very, very simple data model in which we make statements about subjects and objects by linking them with predicates. Okay. So the really, really cool thing about RDF is that individual triples from different resources can be linked to create larger graphs. So here we have two resources with two different triples. These triples can be linked to form a larger graph, providing that they use the same kind of identifiers, the same semantic standards to describe the entities they're talking about. So this makes data integration on the fly possible. We can create a larger graph from individual resources and then ask questions of that graph, which we can't do with the individual data sources. Okay. So again, our goal here is really to make these resources interoperable, standardize their semantics and their data models, create RDF data models for these resources, and then show how we can support the generation of metabolic networks using them as a whole. Okay. So just a couple of results, things we actually did in this study. So we looked at the biological entities in these metabolic network models, and we mapped the concepts in them to a variety of content and schema ontologies. Okay. So then we're using the same descriptors for the same kind of objects in our various resources. So to illustrate one example of this, Ensembl Bacteria is now using ChEBI to describe metabolites, and because ChEBI is used by the Rhea resource, this provides they have a link to the Rhea reactions. So there's now over 22 million Ensembl genes have been mapped to Rhea reactions, and this mapping was generated using a common vocabulary to Ensembl Bacteria and Rhea, which is the enzyme classification. Okay. So in a nutshell, Ensembl Bacteria is now using Rhea, which is using ChEBI. For those of you who don't know, ChEBI provides biochemical reactions with, Rhea is providing biochemical reactions using ChEBI components. We have around 11,000 reactions in this resource. This is just a typical one. All the metabolites within these reactions have as complete structural representation as possible, and they're all balanced for mass and charge. Okay. So we did some semantic standardization as shown here, and then the next step was really to generate interoperable data models. So to generate RDF models which can be combined on the fly for network generation. So here is one of those models. It's the MicroScope data. MicroScope is one of the more complex resources that was included in this implementation study. So ELIXIR France, they created an RDF export for this, which is now available for download at the link given here. So the aim of this webinar is not really to describe the data models in depth. If you're interested in that, you can go and get them, actually. Okay. What I'd like to do in the remaining few minutes is to illustrate a couple of applications that I hope will show you why we chose to use RDF in this implementation study. So there's two main aspects, really. One is to simplify third-party use of our tools, and I'll illustrate that using HAMAP. HAMAP rules as SPARQL. And the second aspect is actually the one which is highlighted previously for RDF, is to simplify the federation of our data on the fly. So to that end, we'll address a couple of the use cases in metabolic network generation using queries that federate different SPARQL endpoints. One of those is mapping enzyme functions to reactions, and another is proposing enzymes for metabolites, sometimes known as gap fill. So if we look at first the development of the SPARQL for HAMAP. So for those of you who don't know, HAMAP is a resource of protein family profiles and annotation rules, so it can be used to classify protein sequences and generate annotations for them. It's created by people doing actually Swiss procuration within the UniProt team, and the rules are used in UniRule for the annotation of UniProt KB Tremble. So it's part of the production pipeline used in UniProt. The profiles themselves are also used in other resources like Interpro to provide protein family classification of their sequences. We also provide actually an annotation service at our website here, for which I give the URL, where you can annotate your proteome. So if you have a proteome in FASTA format or a variety of other formats, you can paste that into the box at our website or upload the file and get it scanned and annotated in SwissProt format, if you like. Okay. Now, although we provide a web-based annotation service, we don't actually provide users, third-party users, with a copy of the pipeline to do HAMAP annotation. Okay. One of the things we looked at in this implementation study is how to translate the logic of HAMAP, where the rules have their own extremely complex format, into something generic, so into SPARQL queries, which can then be run locally by third parties using off-the-shelf software, like Genome, and there's many others. So the idea really is to make it easier to use HAMAP outside of our own infrastructure, so people can use the rules to annotate their own genome and proteome sequence data. So to give you an example, here we have two boxes at the top. We have a user's RDF data, their sequence data, and some classification data. And on the bottom, we have a HAMAP rule which can annotate the sequence. So the user's data contains classification information. The user's particular sequence is a member of a HAMAP family number 300, which is a chorismate synthase. So to use these SPARQL rules, you will need a classification system, but for this you can use something like Interpro scan. So once you have sequences classified with HAMAP, you can then run the annotation rules on them. This is a very simple HAMAP rule below. The construct provides the annotation, so we're going to annotate this particular EC number 4235 for the chorismate synthase. The where clause which follows it includes the conditions under which the annotation applies, and these conditions include not only membership of this HAMAP family, MF300, but also a length constraint, which is known to apply to members of this family in UniProt ChEBI SwissProt. So this provides a very simple demonstration of how you can translate the HAMAP format, which is proper to UniProt, if you like, into something which is generically applicable in SPARQL. Okay. The second reason for using RDF, obviously, is one of its biggest selling points, and that's federating data on the fly. Okay. Again, there's two applications I'd like to showcase here. So during this implementation study, we developed a Rhea RDF data model, but also a SPARQL endpoint, and this is now publicly available. So the queries I'm going to show you, you can try them out yourself, and there's also a range of other queries, demo queries and variants of them, which you can test at this link. Okay. So let's look first at how to map enzyme functions to reactions. This is a common requirement for many users. Okay. So this is a single SPARQL query, and it will allow you to do this. So I'd just like to highlight some of the more salient points. First, we're going to look for Rhea reactions and link them to UniProt proteins via their EC numbers. Okay, so we're going to make a federated query, which utilizes two different SPARQL endpoints. So this service call, it combines the UniProt and Rhea SPARQL endpoints. Okay, so we're beginning at the Rhea SPARQL endpoint, but we're also going to make a call on UniProt, get data from there, and combine it. Okay. So the third part of the query, it specifies that we want UniProt ChEBI SwissProt entries that describe enzymes or proteins of the E. coli reference proteome in UniProt, and this union requires that we identify enzymes where the EC number is annotated either at the level of the complete protein sequence or it's linked to some individual domain within the sequence. So this is a specificity of SwissProt. You can have annotations at multiple levels, and this part of the query is designed to deal with that. Okay, so this is our query. You can copy-paste this into the Rhea SPARQL endpoint and run it if you like. and the result you get back is a set of around 1,600 links between E. coli proteins from UniProt, ChEBI, SwissProt on the left, and Rhea reactions on the right. Okay, and they're linked via their EC numbers. So what we've done is we've joined on the fly the Rhea data via its SPARQL endpoint to the UniProt data via its SPARQL endpoint to generate links which are not present in either of the resources at the current time. Okay. Our last application is something a little bit more novel. It's trying to show how we could use SPARQL endpoints and federated queries to suggest enzymes for metabolites which are lacking reaction at enzyme annotation, sometimes called dead-end metabolites. Now, there's a lot of different algorithms for doing this, many of them based on cheminformatic approaches and sequence homology. These are really the state of the art for this kind of application. We're not suggesting this as an alternative, which could replace those, but it's an interesting supplement which exploits existing features of ChEBI, Rhea, and UniProt to achieve a similar kind of result. Okay, so our metabolite of interest for the purposes of this demonstration is a steroid. It's something called dafachronic acid, which has a role in aging in nematodes, actually. So we might be interested in finding microbial enzymes which could metabolize this, perhaps producing it. So there's no annotated enzymes or reactions for this particular ChEBI entity, either in ChEBI or Rhea, and no mention of this metabolite in UniProt either. So as well as being a dead-end metabolite, it could also be considered a non-starter. Okay. So the SPARQL query that allows us to map this metabolite to potential enzymes is shown here. So again, I'll just show you, I'll highlight some of the salient points about this query. The first thing to note is that we're going to use the ChEBI ontology to actually try and identify enzymes for this metabolite. So this part of the query will retrieve direct parent classes for this metabolite in ChEBI. Okay. So just to illustrate that, our metabolite of interest is this dafachronic acid. If you look at the ChEBI ontology, it's a 3-oxo-delta-1 steroid. Okay, so that's a manual assignment to a ChEBI class that's been made by the ChEBI curators. Okay, so this part of the query allows us to go up the ChEBI ontology and retrieve parent classes. We can also play around with this query, for instance, by adding this plus sign, and that will then take us all the way to the root. Okay, so we have two variants possible, either a simple variant or a more complex variant. For the purposes of this demo, we'll keep it simple, and we'll just stick with the simple variant. We'll only use the parent class. So that's the first thing to note about this reaction. This query is that we're using the ChEBI ontology. This part of the query retrieves all the reactions involving our metabolite of interest. Actually, there are none, but also the parent class to which it belongs, and it maps them to EC numbers. The third part of the query highlighted here combines UniProt and Rhea SPARQL endpoints in one. So again, it's a service call to UniProt, and it's going to use the EC mapping to identify proteins. And again, we get enzymes from UniProt, ChEBI, SwissProt that are annotated with enzymatic activities either at the sequence level, the protein sequence level, or the protein domain level. Okay. So again, you can run this query now on Rhea, and the kind of results you get are shown here. So first we have the ChEBI class for which enzymes were identified, ChEBI 20156, which is our 3-oxo-delta-1 steroid. We get the corresponding EC numbers, the protein counts, which are linked to those EC numbers, the corresponding reaction, and the equation. Okay. So although there are no proteins annotated for this ChEBI metabolite in any of the resources, through this hierarchical traversal, we can identify 530 proteins in UniProt, which could potentially metabolize this particular dafachronic acid. Okay. You can then move to UniProt to explore the results in more detail. 5 of these proteins are found in UniProt, ChEBI, SwissProt from a variety of bacteria, including Microbioal. So just to stress again how this is working, we have a ChEBI metabolite of interest for which we have no annotations. we retrieve chemical classes to which it belongs. We map those to Rhea reactions. So some Rhea reactions include this chemical class. We then link those to UniProt via shared enzyme nomenclature. So this allows us to create links between ChEBI metabolites and UniProt records, which don't exist in any of the resources concerned, and we can do this on the fly. Okay, so that's it for this demonstration. I hope I've shown you that we can federate the existing ELIXIR resources in some implementation study using RDF and SPARQL, and also obviously shared semantics, in order to add collective functionality to these resources that they don't have individually. And we can use that in some real-world situations where annotation is needed for building better computational models of microbial systems. One thing to note is that modeling requires a range of data which is outside the traditional scope of these knowledge bases like Rhea, ChEBI, UniProt, and others, including data like hypothetical reactions, biomass reactions, media composition, and so on. So obviously integrating other specialized resources with these knowledge bases, modeling platforms like MetaNetX and model repositories will really be key to making further progress. So in this regard, I'd like to mention a Microbial Biotechnology workshop which will be held at the ECCB in Athens in September the 8th, on September the 8th. And this will produce a white paper and a detailed roadmap for the future of this particular community. There's a lot of additional partners who have expressed an interest in joining this community, in addition to the few people involved in this initial implementation study. And one of the people most active in this is Jean-Loup Forlon. I'd just like to finish by thanking all the people involved in this study. The people who did the work I've spoken to you about today. So we have Jurgen Bolliman, who worked on HAMAP SPARQL, Thierry Lombardo and Anne Morga, who worked on Rhea RDF and the Rhea SPARQL endpoint, and Nicole Radaski, who led the software development at SIB. Adrien Josso and David Valnet from ELIXIR France, who developed the MicroScope RDF, and Nishadi de Silva, Simon Jupp, Helder Pedro, Kieran Taylor, and Alessandro Rullo at Ensembl EBI, who developed the Ensembl genomes data model for this particular study. I'd also like to thank you for listening to this webinar. Thank you for your attention.